We have a problem. Molecular phylogenies and the fossil record are not reconciled. As a consequence, the timing of early animal evolution is not clear. Hi, my name is Pablo Aragones. I'm a master's student in LIS Lab at the University of Alberta. Today, I'll be talking about the insights to the fossil record that can be gathered from extant sponge morphology. Now, animal evolution corresponds only to the very last fraction of the history of life on Earth, and we do consider the emergence of animals as a major transition in evolution. But as I mentioned, the timing and the details of this event are not fully resolved. The sudden emergence of animals at the base of the Cambrian greatly puzzled Darwin, who argued that eventual fossil discoveries will solve this conundrum. He was right because we now have fossil evidence that push back the origin of animals at least to the last part of the Precambrian, at 555 million years ago. Still, there is rather a large gap with respect to molecular phylogenies that predict the origins of animals at 800 million years ago. The incomplete nature of the fossil record is not to blame, because the conditions at the time were just right to have fossils of exquisite detail. So I'm trying to see and to hear what sponges have to say about this lost record. The main contenders for the sister group of metazoa are Ctenophora and Porifera. There is evidence for both scenarios, and so the debate is not finished. But the traditional view of sponges as the sister group is compelling, since they have a body plan that has not changed since they appeared. They are specialized filter feeders. These provide us with a guideline of what to look for in the rocks. And there have been candidates to fill in this gap. For instance, Otavia antiqua has an interesting timing at 760 million years ago, but its morphology more closely resembles that of a tested amoeba. Eocyatispongia at 600 million years ago exhibit an osculum-like structure, but only one specimen has been reported so far. Now we have Tectardis avalonensis from Mistaken Pond in Newfoundland, Canada that sits at 575 million years ago, and it has gained steam as the long sought-after Precambrian sponge due to its shape and abundance. So remember this name, we'll be talking about it later. First, we need to ask, what is a sponge? So in terms of structure and function, a sponge is a highly specialized biological pump that is organized in an almost fractally branching canal system, leading to and from pump units. Its units are the choanocyte chambers, of course, and inside each chamber we have many choanocytes. This is the pump and filter unit that need to overcome the system pressure loss. So besides spicules, the character of a sponge are the elements of its aquiferous system. We have the incurrent pores of Orostia, the subdermal space, the incurrent and excurrent canals, the choanocyte chambers, the internal cavity or atrium, and the excurrent opening called the osculum. But can we find this in the fossil record? Not really. After fossilization, we are often left with a compression that recovers the external outline of the sponge and not much more. So we need to find a way to compare modern sponge to the fossil record, which is basically just an outline. So because shape is the main information that we can have in fossils, in paleontology it is often assumed that anything tube-shaped could potentially be a sponge, and that is the case for Tectardis. So the premises of this work is that sponges are problematic. What fossilizes are spicules, while soft tissue doesn't preserve well. But we do have an outline to compare fossils and extant forms. Plus, the relationship of modern sponge morphology to the pump is widely reported, but it has never been used to compare to the fossils. So my hypotheses are that modern sponge share a morphometric character that indicates the pump. The osculum size scales with the number of choanocyte chambers, and this with the excurrent speed. Also, the ratio of the osculum area to the surface area should be conserved in sponges, and assuming that the pumping character of a sponge has not changed, we should find this relation in the fossils as well. So in terms of methods, I created a dataset from previous lab work and unpublished images, and also I took some measurements of my own. Some of the species I work with are Tetia californiana from Banfield and Gioria barreti from Norway. These are very spherical sponge, as you can see, and Caldispongia vaginalis, that is a very cylindrical sponge. So the surface area was easy to obtain as these resembled very ideal shapes. Other irregular shapes like the encrusting sponge from Banfield Haliclona, Mollis, and Neopetrosia problematica were a little bit difficult to work with. They posed a bit of a challenge. But the surface area could still be obtained through a combination of polygons. I also conducted a meta-analysis to get data from fossils. I compared two extremes of the Phanerozoic, the Cambrian period at the beginning and the Eocene period almost toward the end. For modern sponge, I used the scanning electron micrographs to count the quanosite chambers, and I used the software ImageJ to measure the diameter of the canals, the quanosite chambers, and estimate the osculum area and surface area. In terms of the statistical analysis, I perform a Spearman-Wright correlation on the barbels to get the relations of osculum area to the screw speed, the volumetric flow rate, and the number of quanosite chambers. I perform a two-sample t-test assuming unequal variances to the osculum to surface area ratio, and then I analyze the slope of the ratio using an analysis of covariance. Some of the variables of interest to compare are the osculum area and the surface area. And then we have the speed at the osculum or screw speed, and with the osculum area and the screw speed we can calculate the volumetric flow rate. We can also have the number of quanosite chambers per unit volume from the scanning electron micrographs, and we can extrapolate that value to get the total number of quanosite chambers in a sponge. So the first results of this work involve the scaling of the morphology of the pump. Here we can see in figure A the surface area on the x-axis and the osculum area on the y-axis. We do expect to have this relation because the larger sponge is, the larger the osculum will be, but we are trying to describe the slope of this relation. And we can see this gross morphology and how it scales with the volumetric flow rate. So here we have the surface area ratio, the surface area, and then we have the flow rate. So that's for surface area, and we can also plot the osculum area to the x current speed. The thing that is interesting is that when you have the ratio, so the osculum to surface area ratio, you have a negative relation with the x current speed. So the bigger the ratio is, the slower the sponge pump. So to understand the osculum to surface area ratio more intuitively, here I have two pictures. On top, the Extant sponge Haliclona mollis from Banfield, and on the bottom, the fossil sponges from the Cambrian Bauxia gracilenta. First, we take the linear dimensions of the outline and calculate the surface area and the osculum area. Then we just divide the osculum area to the surface area. The proportion of these two areas is the osculum to surface area ratio. And when we plot this, we see some interesting patterns. For example, we see that in modern sponges we have a narrow range. They all converge to about 0.01. On the other hand, fossils have a wider range. And if we look to the right, Tectartes, even though it is far away from modern sponges, it does lie within the range of some Cambrian sponge general like Hamptonia and Haciella, and also the Calcarean sponge, a feldspongea, also from the Cambrian. So Tectartes do have a similar ratio that is that of other known sponges. Now let's compare the slopes. If we plot the surface area to osculum area and compare the slopes, we can see that fossil and modern demosponges share a slope. So this is not statistically different until using the analysis of covariance. If we compare fossil and modern Exactinolida, we find the same thing. So these slopes are the same, which indicates that there is a class-specific slope, and we can prove that using the test from demosponge and hexactinolids. So we see that the slopes are different. Now, even more interesting is when we compare the demosponges to Tectartes. We have that it is the same slope. So Tectatis has the ratio of some sponge genera, and it also has the same slope as the demo sponges. So in conclusion, although some sponges are asymmetrical, they do have a body ratio that relates to the bump character. This is the osculum area to surface area ratio. And the slope of the osculum to surface area seems to be characteristic in modern sponges. This ratio can be applied to modern sponges and fossil forms to compare them, and the slope can discriminate between classes and give valuable insight into the putative Precambrian sponge fossil record. Additionally, it can be used to estimate the pumping rate of ancient sponge reef as those of the Cambrian. Going back to the putative sponge Tectatis abalonensis, it displays the same morphometric characters as the rest of the demo sponges. I would like to take the opportunity to thank Dr. Salilis, my supervisor, and Nikita Sergenko, my lab mate, who also helped me collect the sponges, and also the dive crew at Banfield Marine Science Centers, and the MIDAX Graduate Fellowship for supporting in the first part of my master, and the whole LIS lab for all the invaluable feedback. And I would also like to thank all of you for listening to my talk, and I'll be happy to take any questions.